I want you to try something for me. Just close your eyes for a second if you can. Picture yourself standing right in the middle of a dense, humid forest.
Setting a scene today. I like it.
Yeah, we're going immersive. So you're in this forest. It's messy. There's mud caking the bottom of your boots. You've got these massive ancient trees blocking out the sky above you. Maybe a squirrel darts across a branch, or, you swat a mosquito off your arm.
Sounds pretty standard. Just nature doing its thing.
Exactly. It looks like just nature. But imagine you had a pair of magic glasses that could let you see absolutely every single living thing in that frame. What would you actually be looking at?
Oh, well, those glasses.
Yeah.
Chaos. Absolute unmitigated biological chaos. I mean,
Yeah.
You wouldn't just be looking at the trees and that squirrel anymore. You'd see fungi digesting a fallen log, breaking down lignin molecule by molecule.
Right.
You'd see billions, literally billions of bacteria in the soil fighting this invisible chemical war against other bacteria. And you'd see viruses just floating in the air, these inert little crystals just waiting for a lung to land in.
It's essentially a biological jungle, but on a microscopic scale.
It really is. And the thing about human beings, our brains, we absolutely hate chaos. We have this desperate evolutionary need to sort things out.
We need to categorize.
We do. We need to be able to look at the world and say, this is safe, that is dangerous, this is food, that is poison, this is a cat, that is a dog.
We need boxes. We need to put things into neat little categories so we don't just completely lose our minds.
That is exactly it. We need boxes. And that is exactly what we are doing today on this deep dive. We are looking at one of the most ambitious intellectual projects in human history, which is biological classification.
Now, before you roll your eyes and check the runtime on this, thinking, oh, great, an hour of Latin names and dusty lists, let me stop you right there. Because if you are listening to this, you are very likely staring down the barrel of the Neet exam.
Oh, yeah. And for the Neet aspirant, Chapter 2 of the NCERT textbook, Biological Classification, it isn't just a chapter you can skim, it is a minefield.
A minefield. That sounds a little ominous, I'm not going to lie.
Well, it absolutely is. It's incredibly dense. It's packed with very specific, rigid rules. And then, and this is honestly the worst part for students, it is packed with organisms that completely break those rules.
The exceptions.
Yes, the exceptions. And the exceptions are exactly where the examiners live. They absolutely love to ask you about the weirdos, the things that just do not fit the mold.
Like all bacteria have cell walls except which one?
Exactly. Or all fungi have chitin except which class, that kind of thing.
Right. So here is our mission for today. We aren't just going to read you the list. That would be boring and frankly useless. We are going to decode the actual logic behind it. We want to help you understand why R.H. Whittaker drew the specific lines where he did. We're going to really look closely at the big three: Monera, Protista, fungi. And we're even going to argue a bit about whether a virus is actually technically alive.
And we're going to do all of that by looking at the systems themselves, not just the vocabulary. Because if you can understand the underlying evolutionary logic, you don't actually have to memorize as much. The answers just they just click into place.
That is the promise today. High yield, deep understanding of the source material. So let's go back, way back, before we had microscopes, before we had any clue what DNA was. How did humanity even start organizing this mess?
Well, the very first serious attempt, and I should say scientific attempt, was Aristotle. And you really have to give the guy some credit here, because he was working with the ultimate limitation.
Which was just his bare eyes, right?
Exactly. No lenses, no microscopes, nothing. So his classification system was purely morphological. It was entirely based on, what does this thing look like?
Keeping it simple.
Brutally simple. For plants, he literally just looked at their size and their basic structure.
Yeah.
So he asked, are you a giant tree? Are you a medium-sized shrub? Or are you a tiny herb? And that was it.
Okay. Fair enough. And for animals?
This is honestly my favorite Aristotle dichotomy. He divided all animals into two distinct groups, those with red blood and those without.
Red blood versus no red blood.
I know it sounds kind of funny to us now, but think about his perspective. He's cutting things open to see how they work. He sees a cow, it bleeds a red fluid, he squishes a beetle. It's kind of a clear or yellowish fluid. It's a very straightforward binary choice.
I mean, it works if you're a butcher, I guess, but it completely fails if you're trying to be a biologist.
Right, because it entirely ignores everything internal. It ignores how these creatures reproduce. It ignores their cellular makeup, their evolutionary history, everything. But amazingly, it stood for a very long time.
Until Linnaeus comes along.
Yes. Enter Linnaeus, the father of taxonomy. He is the one who gives us the two-kingdom system.
Plantae and Animalia.
The classic binary we all pretty much grow up with. The rule is simple. If it moves around and eats things, it's an animal. If it stays put and is green, it's a plant.
It feels very intuitive.
It does feel intuitive. But for a modern microbiologist, this system was an absolute disaster. It was basically a lie.
Why? What did Linnaeus get so fundamentally wrong?
He lumped things together that had absolutely no business being in the same room, let alone the same biological category. And understanding exactly why this failed is a classic, super high-yield concept for the exam. You really need to know the three big contradictions of the two-kingdom system.
Okay, let's break this down for everyone listening. What is the very first big mistake he made?
He didn't distinguish between prokaryotes and eukaryotes?
Let's pause right there. Just for the listener who maybe hasn't opened their textbook in a week or two, let's reestablish that baseline. Can you define that difference?
Sure. A eukaryote keeps its genetic material, its DNA, safely locked inside a nucleus. It has a vault. It's highly organized.
And a prokaryote.
A prokaryote. Its DNA is just sort of floating around freely in the cytoplasm. It's messy. It's a much more primitive setup.
So Linnaeus took bacteria, which are these tiny, primitive, completely nucleusless things, and he put them in the exact same group as what?
As an oak tree or a rose bush. These massively complex, multicellular eukaryotes. He threw them all in the same box simply because they both possessed a cell wall.
Wow. That's like putting a bicycle and a Ferrari in the exact same category just because they both happen to have tires.
That is a perfect analogy.
Yeah.
It ignores the engine entirely. So that is mistake number one. Mistake number two is that he lumped unicellular and multicellular organisms together.
The source material specifically mentions Chlamydomonas and Spirogyra here.
Yes, those are the classic examples to remember. Chlamydomonas is a tiny single-celled alga. It swims around, it does its own individual thing. Spirogyra, on the other hand, is a long filamentous multicellular organism.
But in the two-kingdom system...
In that system, they are both just plants. They're neighbors in the same exact category.
Okay, and the third mistake, I think this is the one about food, right?
Yes, it is. It's the distinction between photosynthetic and non-photosynthetic organisms. Linnaeus put fungi, which literally eat decaying organic matter, they are heterotrophs, into the exact same group as green plants that manufacture their own food using sunlight.
See, I've always found that one so weird. Why did anyone ever think a mushroom was a plant? It doesn't look like a plant. It's fleshy. It doesn't have any leaves. It's not green.
It really all comes down to one single feature again, the cell wall.
Ah, the wall.
Linnaeus looked at a plant cell under an early lens, and it had a rigid wall. He looked at a fungal cell, and it also had a rigid wall. So he basically just said, well, close enough. You're a plant.
But those walls aren't actually the same, are they?
They absolutely are not. And this right here is a crucial nuance you need for the exam. Plants have cell walls made of cellulose. Fungi have cell walls made of chitin.
Chitin. Wait, isn't that the exact same stuff that insect exoskeletons are made of?
Precisely. So chemically speaking, a mushroom's armor is much more similar to a beetle's shell than it is to a tree's bark.
That's wild.
But Linnaeus couldn't see the chemistry. He didn't have the tools. He just saw the rigid structure of the wall.
Right. So the two-kingdom system is essentially falling apart under scrutiny. It's lumping totally incompatible things together. Biology needed a much better system. And this brings us to the hero of Chapter 2, R.H. Whittaker.
1969. That is a date you absolutely want to remember. 1969 is when Whittaker proposes the five-kingdom classification.
Monera, Protista, Fungi, Plantae, and Animalia.
This is the gold standard. And for your exam, you need to know the specific criteria that Whittaker used to make these divisions. It wasn't just random guessing. He used 5 main pillars to build this entire system.
All right, I'll rattle them off one by one, and you tell me why they matter so much. Number 1 is cell structure.
Prokaryotic versus eukaryotic. This is the primary foundational divide. This isolates Monera from absolutely everything else, and it completely fixes Linnaeus's first big mistake.
Got it. Number 2 is body organization.
Is the organism unicellular?
Yeah.
Is it multicellular? Does it actually have differentiated tissues? This criterion fixes the whole Chlamydomonas versus Spirogyra problem.
Number 3, mode of nutrition.
This one is huge. Autotrophic versus heterotrophic. This is the rule that finally, officially split the fungi away from the plants.
Number 4 is reproduction.
Simply looking at how they propagate. Is it a sexual process or an asexual one?
And finally, number 5, phylogenetic relationships.
Evolutionary history. Who is actually related to whom? Whittaker tried his best to make the classification system actually reflect the true family tree of life on Earth.
Okay, so we have the map laid out. Now let's actually drive through the territory. We are going to start right at the foundation. Kingdom Monera.
Kingdom Monera, the bacteria.
Now, the source text says that bacteria are the sole members of this kingdom and that they are cosmopolitan, they are everywhere.
They truly are the most abundant microorganisms on the planet. You can find hundreds of them in just a single handful of ordinary backyard soil. They thrive in boiling hot springs, in freezing deserts, in the deep ocean trenches, and obviously inside other organisms. They are the ultimate biological survivors.
And even though their physical structure is technically very simple, they're prokaryotic, no nucleus, their behavior is actually incredibly complex, specifically their metabolic diversity.
Yes, that is the key phrase to lock in. Extensive metabolic diversity.
What does that actually mean in plain English?
It basically means they have the most varied menu of any life form on the planet. If you and I want energy, we have to consume organic matter. We are very limited. Bacteria. Some of them use direct sunlight, some use hydrogen sulfide, some literally eat iron, some process nitrogen, some are parasites that feed off us. They can figure out how to do it all.
Okay, let's talk about their shapes really quickly. This is a very common visual check on exams. Bacteria are grouped into 4 main categories based strictly on their shape.
Right. You have the spherical ones, which are called coccus. You have the rod-shaped ones called bacillus.
They look kind of like a tiny pill capsule.
Exactly like a pill capsule. Then you have the comma-shaped ones, vibrium, and finally the spiral-shaped ones, which are called spirillum.
Simple enough. But Monera isn't just one big, happy, identical family. We have to split it into 2 major subgroups. And this is exactly where the exam questions start getting very technical. We have the archaebacteria and the eubacteria.
Let's start with the archaebacteria. These are the ancient bacteria. I like to think of them as the special forces of the microbial world.
Why special forces?
Because they willingly go where absolutely no other life can survive. They are the extremophiles.
The source lists 3 specific types of extremophiles you need to know. First, we have the halophiles.
The salt lovers. Imagine a place like the Dead Sea. The water is so incredibly salty that it literally sucks the moisture out of normal cells through osmosis. But halophiles, they absolutely thrive there.
Then we have thermoacidophiles.
Heat and acid lovers. Think of the boiling hot springs in Yellowstone. You're dealing with temperatures that would hard boil an egg and a level of acidity that would literally dissolve metal.
And finally, the methanogens.
These are typically found in marshy, swampy areas, but crucially, they're also found in the gut of ruminant animals, so cows and buffaloes. They're the ones responsible for producing methane or biogas from the dung of these animals.
Now, here is the million-dollar exam question. Why? What exactly makes archaebacteria tough enough to survive living in a boiling hot spring?
It is the cell wall. It is always the wall. Archaebacteria have a fundamentally different cell wall structure compared to all other bacteria.
How is it different chemically speaking?
Well, normal bacteria have a cell wall made of a substance called peptidoglycan. Archaea do not. But even more importantly, their cell membranes contain branched-chain lipids.
Branched-chain lipids.
Yes. This branching structural formation makes the membrane incredibly stable and highly heat-resistant. It's basically like wearing a military-grade Kevlar vest while every other organism is just wearing a cotton t-shirt.
That's a great visual. Okay, moving on to the true bacteria, the eubacteria.
These are characterized by having a very rigid cell wall, and if they happen to be motile, they have a flagellum to help them swim.
Within the eubacteria, we have a very important group called the cyanobacteria, also commonly known as blue-green algae.
Which I have to point out is a completely terrible name.
Really? Why is that?
Because it constantly confuses students. It literally has the word algae right there in the name, so people automatically think, oh, it must be a protist or a plant. But it is absolutely a bacteria. It is a prokaryote. It squarely belongs in Kingdom Monera.
So if you see blue-green algae on the test paper, you instantly scream Monera.
You scream Monera in your head, yes. But they are similar to plants in one very specific way. They possess chlorophylla. They are photosynthetic autotrophs. They can manufacture their own food using sunlight.
And the source material mentions that they very often have a gelatinous sheath surrounding their colonies.
Right, which is exactly what creates those massive blooms you see in highly polluted bodies of water. You know, that thick, nasty green scum on a stagnant pond? That is a cyanobacteria bloom.
Now, there is a very specific structural feature in cyanobacteria that is an absolute favorite for examiners, the heterocyst.
Ah, yes. The heterocyst. This is a truly perfect example of biological ingenuity. See, cyanobacteria need to fix nitrogen from the atmosphere. but the specific enzyme required to fix nitrogen, which is called nitrogenase, is actually destroyed by the presence of oxygen.
Well, that seems like a massive problem if you are a photosynthetic organism that actively produces oxygen as a byproduct.
It's a huge problem. You are essentially poisoning your own internal machinery. So what do they do to solve this? They build a bunker. A bunker. That is exactly what a heterocyst is. It's a highly specialized, very thick-walled cell that is designed to keep all oxygen out. Inside that protective bunker, the bacteria can safely fix nitrogen, while the neighboring cells on the outside continue doing the photosynthesis.
That's incredible. It's an internal division of labor.
Inside a single-celled organism colony. It's brilliant. Nostoc and Anabaena are the two classic examples you need to know here.
So Nostoc and Anabaena equals heterocysts equals nitrogen fixation. Make sure you connect those dots.
Definitely. Then moving on, you have the chemosynthetic bacteria. These guys are basically the recyclers of the microbial world. They oxidize inorganic substances like nitrates, nitrites, and ammonia to produce energy for ATP. They play a truly massive role in recycling essential nutrients like nitrogen, phosphorus, iron, and sulfur back into the ecosystem.
And finally, we have the heterotrophic bacteria, the most abundant ones overall.
These are mostly decomposers. And this is really where we see the biggest direct impact on human life. They are the ones that help make curd from milk, specifically lactobacillus. They help us produce antibiotics, and they fix nitrogen in the roots of legume plants.
But they definitely also have a dark side.
They absolutely do. The pathogens, cholera, typhoid, tetanus, citrus canker, every single one of those is a bacterial disease.
Listener, make a mental note of that specific list. Cholera, typhoid, tetanus, citrus canker. If you get a multiple choice question asking which disease is viral and one of these is an option, you can immediately cross it out.
Good tip. Now, before we finally leave Kingdom Monera, we absolutely have to mention the major exception, the outlier of the group. Mycoplasma.
The joker in the pack.
Mycoplasma are fascinating because they are organisms that completely lack a cell wall.
Wait, no cell wall at all? But I thought we established that was one of the key defining features of bacteria.
Exactly, which is why it's such an important exception. And because they don't have a wall, they are naturally resistant to certain antibiotics that specifically target cell wall synthesis, like penicillin.
Because penicillin works by stopping the wall from being built in the first place.
Right, so if there's no wall to begin with, the drug literally has nothing to attack. It's useless.
That is incredibly sneaky.
They are also recognized as the smallest living cells known to science. And, this is a very crucial detail for the exam, they can survive entirely without oxygen.
So they are anaerobic, wall-less, incredibly tiny, and very often pathogenic to both plants and animals. That is your profile for Mycoplasma.
And that pretty much wraps up Monera. Now, we are going to cross a boundary into the much more complex eukaryotic world, Kingdom Protista.
Kingdom Protista. The source text actually has a really interesting, slightly vague line about this kingdom. It says, the boundaries of this kingdom are not well defined.
I always call it the miscellaneous drawer of the biological kitchen.
Explain that analogy.
Well, if a scientist finds a single-celled organism that clearly has a nucleus, meaning it's definitely a eukaryote, but it isn't clearly a fungus, and it isn't a plant, and it isn't an animal, you basically just throw it into Protista.
Yeah.
It acts as a catch-all. It forms this evolutionary link between Monera and the three higher complex kingdoms.
Which means it's a wildly diverse group of organisms. We have 5 specific groups to get through here. Let's jump in. Group 1, the crucifites.
This group includes the diatoms and the golden algae, which are also called desmides. You'll find them in both freshwater and marine environments.
I really want to focus on diatoms. The source uses a very memorable, specific analogy for how their cell walls are constructed.
A soapbox.
A soapbox, like for travel.
Exactly like that.
Yeah.
Think about those old plastic travel cases you'd put a bar of soap in. You have a bottom half, and you have a slightly larger top half, and they overlap to fit really tightly together. That is the exact architectural design a diatom uses to build its wall.
And what exactly is this overlapping wall made of?
Silica.
Silica, like sand, like glass.
Basically, yes. The cell walls are heavily embedded with silica. This makes them practically indestructible. They're essentially living inside tiny, microscopic glass boxes.
So what actually happens when they die?
The inner organic part of the cell rots away, but the outer glass box remains completely intact. It sinks and falls to the ocean floor. And over billions of years, this massive accumulation of shells forms what we call diatomaceous earth.
That gritty white powder.
Yes. And because it's so gritty and abrasive, we mine it and use it for polishing. And because it's incredibly porous, we use it industrially for the filtration of oils and syrups.
They are ecologically super important too, aren't they?
Massively. They are the chief producers in the oceans. They form the absolute base of the marine food web. So they're just tiny little soapboxes, but they have a colossal global impact.
Group 2 in Protista, the dinoflagellates.
These are mostly marine and photosynthetic. They can actually appear in a wide variety of colors: yellow, green, brown, blue, red. It all just depends on the specific main pigments present in their cells.
What about their physical structure?
They have very stiff cellulose plates on their outer surface. Under a microscope, they almost look like they're wearing medieval plate armor. And the name itself, dinoflagellate, gives you a big structural clue. They usually have two flagellus.
Two of them?
Yes. One is longitudinal, running down the length of the cell, and the other is transverse, running across it, usually in a little furrow between the wall plates. This unique arrangement actually makes them spin like a top as they propel themselves to the water.
And what is the major high-yield example here?
The organism responsible for red tides, Gonielax. These specific red dinoflagellates can multiply so incredibly rapidly that they actually make massive stretches of the sea appear deep red.
Is that just a visual thing, or is it dangerous?
Oh, it is extremely dangerous.
Yeah.
They release powerful toxins into the water that can kill huge numbers of marine animals, including fish. So a red tide isn't just a pretty color change, it's a massive toxic ecological event.
Gonielax causes toxic red tides.
Got it.
Okay, group 3, the euglenoids.
These are fascinating little guys because they basically suffer from a biological identity crisis. They bridge the gap between plants and animals. They're mostly found in fresh water, specifically in stagnant water.
No, do they have a cell wall?
No, and this is key for the exam. No cell wall. Instead, they have a specialized protein-rich layer called a pellicle.
A pellicle? What is the advantage of that?
It makes their body incredibly flexible. A traditional cell wall is very rigid. It locks you in. A pellicle allows them to bend, squish, and wiggle through their environment.
And what about that plant-animal identity crisis you mentioned?
Well, they have chloroplasts, and they are fully photosynthetic in the presence of sunlight, so behaving exactly like a plant. But if you deprive them of sunlight, say night falls or they sink into muddy water, they flip a switch. They instantly start behaving like heterotrophs, actively predating on smaller organisms, exactly like an animal.
So they are mixotrophic, they can do both.
Exactly. And what's really interesting is that their photosynthetic pigments are entirely identical to those found in much higher complex plants. The classic textbook example here is, of course, Euglena.
Group 4, the slime molds.
These are saprophytic protists, meaning they survive by feeding on dead and decaying organic matter. They slowly move along, decaying twigs and fallen leaves, just engulfing organic material as they go.
The life cycle here is the trickiest part. It changes dramatically based on their environment, kind of like a Dr. Jekyll and Mr. Hyde situation.
Right. So under suitable conditions, meaning there's plenty of food and moisture available, they all aggregate together. They literally merge to form a giant multinucleated slimy mass called a plasmodium. And this thing can grow and spread over several feet.
It's just one big creeping community party.
But when things get tough, when conditions become unfavorable, like it gets too dry or food runs out,
a party abruptly ends.
Exactly. The plasmodium differentiates, it changes its structure entirely, and forms fruiting bodies that bear spores right at their tips.
And these spores are survivors.
They're incredibly tough. They possess true, rigid walls. They are highly resistant to extreme temperatures and prolonged drought, and they can actually survive in a dormant state for many years. When the time is right, they are dispersed by air currents to find a better home.
So to summarize slime molds for the listener, happy conditions equals plasmodium, the giant slimy mass. Unhappy conditions equals fruiting bodies and tough spores with true walls.
That is the perfect way to remember it.
All right, finally, group 5 of Protista, the protozoans.
These are the primitive evolutionary relatives of animals. They are all heterotrophs, living either as active predators or as parasites. We generally divide them into 4 subgroups based entirely on how they move around.
4 movement types. Let's go through them. First up.
First are the amoeboid protozoans. They move and capture their prey using pseudopodia, which translates to false feet. This is exactly what the amoeba does. The marine forms of these often have silica shells, and some of them, like entamoeba, are serious human parasites.
Second group.
The flagellated protozoans, as the name implies, they have flagella for movement. The highest yield example here is trypanosoma, which is the parasite that causes the disease sleeping sickness.
Third.
The ciliated protozoans. These organisms are covered in thousands of tiny, hair-like cilia that all beat in precise coordination, kind of like a massive rowing team on a boat. They also feature a cavity, called the gullet, that opens to the outside of the cell surface. The classic lab example is Paramecium.
And the fourth and final group,
The sporozoans. These are unique because they have an infectious, spore-like stage in their life cycle. And the absolute most notorious one you must know is Plasmodium.
Wait a second. Plasmodium. Didn't we literally just talk about Plasmodium when we were covering slime molds?
Excellent catch. And this right here is a classic intentional confusion point on exams. This is a massive red flag for the listener to pay attention to.
Please explain the difference clearly so nobody gets tripped up.
In the context of slime molds, the word Plasmodium is simply the common descriptive name for that aggregated, slimy body structure. It describes the physical mass. But in the context of protozoans, Plasmodium, with a capital P, is the actual genus name of a specific organism. It's the malarial parasite.
That is a crucial distinction. Plasmodium, the protozoan parasite, causes malaria, which obviously has a staggering, devastating effect on human populations worldwide.
Exactly. Do not ever mix those two up on a test. One is just a harmless pile of slime on a log. The other is one of the deadliest infectious parasites in human history.
All right, that officially covers Protista. Now let's leave the water behind and head to the damp forest floor. We are moving to a kingdom that absolutely loves warm, humid places, the kingdom fungi.
Fungi are wonderfully unique. They're heterotrophic, but they have a very specific, totally alien physical structure compared to us.
Let's try to visualize this. When we walk through the woods and we see a mushroom popping up from the dirt, we are really only seeing the fruit, right? What is actually going on underground?
The network. That's what's underground. With the sole exception of yeast, which is unicellular, fungi are filamentous organisms. Their bodies are constructed of these incredibly long, slender, thread-like structures called hyphae.
They look kind of like microscopic cables.
Yes. And the massive tangled web of all these cables working together is called a mycelium. Think of the hidden fiber optic internet cables running under a major city. That network is the mycelium. The mushroom is just the Wi-Fi router sitting on the desk.
I love that. Now, there are two different ways these hyphae cables can be built, and this is vocabulary that will definitely show up on the exam.
Right. So imagine a very long pipe. In some types of fungi, the pipe is completely continuous. It's just one huge, long, undivided tube filled with cytoplasm, and it has thousands of nuclei just floating around freely inside it.
And those are called coenocytic hyphae.
Exactly. Continuous tube, multinucleated. Coenocytic. In other types of fungi, that long pipe actually has physical dividers or cross walls built at regular intervals. These internal walls are called septae.
So those ones are septate hyphae. And what about the fungal cell wall itself? We touched on this earlier with Linnaeus' mistake.
Chitin and polysaccharides. Not cellulose like you see in plants, to chitin. This is the absolute defining chemical signature of the entire fungal kingdom.
Let's talk about how they reproduce, because fungi are unbelievably prolific. They can utilize vegetative, asexual, and sexual reproduction.
Vegetative reproduction happens through fragmentation, fission, or budding. Asexual reproduction primarily involves producing vast numbers of spores, things like conidia, superangiospores, or zoospores.
But the sexual cycle is where the specific steps really matter for the test. It's not just a simple matter of sperm meets egg. There are three very distinct biological steps.
Step 1 is plasmogamy. This is the physical fusion of the protoplasm between two motile or non-motile gametes. The actual cells merge together into one.
Okay, step 2. Karyogamy.
Karyogamy is the subsequent fusion of the two distinct nuclei inside that newly merged cell.
And step 3 is meiosis.
Once the nuclei fuse, the resulting diploid zygote undergoes reduction division, or meiosis, to eventually form new haploid spores.
Plasmogamy, karyogamy, meiosis, PKM. Memorize that order. Now, usually in a standard cycle, step 1 leads immediately right into step 2, right?
Right. And the lower, more primitive fungi, like the phycomycetes, the cells fuse, the nuclei instantly fuse right after, and boom, you have a diploid organism. But in the higher, more advanced fungi, specifically the ascomycetes and the basidiomycetes, they actually hit a pause button.
A pause button.
Yeah, plasmogamy happens. The two cells successfully merge their cytoplasm, but the two nuclei inside, they don't actually fuse immediately. They just sit there side by side, sharing the same cellular space.
So you temporarily have one single cell containing two separate nuclei.
Exactly. This is known as an N+N stage. Specific cell is called a dekaryon. And this entire delayed phase of their life cycle is called the dikaryophase.
It's basically like a really long engagement period before the actual marriage happens.
That is a very accurate way to put it. Eventually, down the line, they finally do fuse, completing karyogamy, and then they immediately undergo meiosis. But the existence of that prolonged dokaryophase is a highly unique defining feature of the Acomycetes and Basidiomycetes.
Okay, we have 4 major classes of fungi to get through now, and these are categorized mainly by the morphology of their mycelium and exactly how they go about producing their spores.
Class 1, the Phycomycetes. These are generally considered the lower fungi.
Where are we mostly finding these?
Mostly in aquatic habitats, on damp, decaying wood in very moist places, or acting as obligate parasites on living plants.
And their structure.
Their mycelium is aseptate and coanecytic, which logically makes sense, right? They are the most primitive group, so they have that simpler continuous tube structure without the complex cross walls.
What about their reproduction?
Asexual reproduction happens via zoospores, which are motile, or aplanospores, which are non-motile. And importantly, these spores are produced endogenously inside a specialized sac called a sporangium.
Let's define that term clearly. Endogenous means they are produced on the inside, kind of like peas growing inside a pod.
Exactly. Endogenous equals inside.
Any specific examples we need to memorize?
Mucor, Rhizopus, which is the very common black bread mold you'll eventually find in your kitchen if you leave bread out, and albugo, which is a parasitic fungus that specifically attacks mustard plants.
Okay, moving to class 2, the Ascomycetes, commonly known as the sac fungi.
These are mostly complex and multicellular, like penicillium, but this group also weirdly includes the unicellular yeast, Saccharomyces. In terms of lifestyle, they could be saprophytic decomposers, parasitic, or, interestingly, coprophilous.
Coprophilous, that's a fun word. What does that mean?
It means they grow exclusively on animal dung.
Yeah, good to know. And what is their structure like?
They have a branched and septate mycelium, so we have officially evolved cross walls now. The continuous tube era is over.
Now let's talk about the spores for Ascomycetes, because this is where the terminology gets genuinely confusing.
It does. Their asexual spores are called conidia, and these are produced exogenously on specialized structures of the mycelium called conidiophores.
Exogenous means they are produced on the outside, exposed, like dust clinging to the outside of a stick.
Right. But their sexual spores, which are called ascospores, are produced endogenously, safely, inside little sac-like structures called asci.
Okay, listener, pay close attention. Conidia, the asexual ones, are exogenous. Ascospores, the sexual ones, are endogenous. That is a massive trap just waiting to happen on a multiple choice test.
It absolutely is. Some key examples for this class include Aspergillus, Claviceps, and Neurospora.
Why is Neurospora so famous in biology?
Because it is used extensively worldwide in biochemical and genetic lab work. It is essentially the fruit fly of the fungal world for researchers. Also, as a fun fact, this exact group includes morels and truffles, which are incredibly expensive, edible culinary delicacies.
Quite a diverse group. Class 3, the Basidiomycetes. These are your classic mushrooms, bracket fungi, puffballs, the really obvious ones we see when hiking in the woods.
They primarily grow in soil, directly on decaying logs and tree stumps, and also as harmful plant parasites, specifically the rusts and smuts. Their mycelium is, again, branched and septate.
How do they handle reproduction?
This is where they get weird. Asexual spores are generally completely absent in this class. They just don't really do the conidia thing. However, simple vegetative reproduction by fragmentation of the mycelium is incredibly common.
And what about sexual reproduction?
They actually do not possess distinct sex organs. There are no specialized male or female parts. Instead, plasmogamy occurs simply by the random fusion of 2 ordinary vegetative somatic cells of different strains that happen to bump into each other in the soil.
Just 2 regular body cells bump into each other and decide to fuse.
Basically, yes. The resultant structure from that fusion is dikaryotic, that N+N stage we discussed, which eventually grows and gives rise to a structure called a basidium. That basidium is actually the microscopic club-shaped structure lining the gills underneath the cap of a mushroom.
And what happens inside that basidium?
Karyogamy, the fusing of the nuclei, and meiosis finally happen inside that basidium, which results in the production of 4 haploid basidiospores. And crucially, these spores are produced exogenously on the very outside tip of the basidium.
Okay, let's do a rapid-fire review of the spore locations, because I know this is a sticky point. Phycomyces spores.
Endogenous, inside.
Ascomyces sexual spores.
Endogenous, inside the sac.
Basidiomyces sexual spores.
Exogenous, outside on the basidium.
Perfect. And the examples for Basidiomycetes?
Agaricus, which is the common button mushroom you buy at the grocery store.
Oh.
Ustulago, the smut fungus. And Puccinia, the rust fungus.
Finally, we arrive at class 4, the Deuteromycetes, better known as the imperfect fungi.
I always refer to this class as the cold case files bin of mycology. They are officially called imperfect simply because we only currently know about their asexual or vegetative phases. We have literally never observed their sexual reproduction stage.
So what exactly happens if a researcher suddenly discovers their sexual phase in a lab one day?
They get immediately evicted. Once we figure out how they actually have sex, they're almost always reclassified and moved into either ascomycetes or basidiomycetes, depending on the mechanism. They graduate out of the imperfect group.
But until that happens, how do they manage to reproduce?
Strictly by producing asexual spores, known as conidia. Their underlying mycelial structure is septate and branched, just like the higher fungi.
And what is their ecological role?
A huge number of them are vital decomposers of leaf litter and plant matter, meaning they help massively in the mineral cycling of forests. Key examples to know are Alternaria, Colloidtotricum, and Trichoderma.
Trichoderma. I've definitely seen that one pop up in past exam papers. Okay, so that thoroughly covers the fungi. An incredibly complex kingdom made of vast networks and millions of spores.
It is a lot to memorize, but the structural logic is definitely there.
Now, the syllabus does briefly mention Kingdom Plantae and Kingdom Animalia here at the end, but the source text explicitly notes that these are covered in much greater exhaustive detail in completely separate dedicated chapters later on. So we are going to skip the deep dive on those for today. Just remember the very foundational basics. Plants have cellulose cell walls and use chlorophyll for photosynthesis. Animals are multicellular, have absolutely no cell walls, and eat other things for energy.
Simple enough for now, but now we have to talk about the twilight zone. The biological entities that Whittaker intentionally left entirely out of his beautiful five-kingdom system.
The unclassified, the acellular anomalies, viruses, viroids, prions, and lichens.
This is exactly where biology suddenly gets very philosophical. Whittaker did not include viruses in his five kingdoms, and you have to understand why.
Simply because they aren't actually considered truly living organisms.
Right. If you rigidly define living as possessing a functional cellular structure capable of independent metabolism, then viruses are dead. They are non-cellular. Outside of a living host cell, they are just inert, crystalline structures. They're essentially just very complex rogue chemicals.
But the second they actually get inside a host...
Oh, then it's a completely different story. Once inside, they completely hijack the host cell's internal machinery to furiously replicate themselves, which inevitably ends up killing the host. They are the ultimate obligate parasites.
The historical timeline of virology is actually a very frequent target for exam questions. It plays out almost like a historical detective story spanning decades. We have 4 major names you need to commit to memory.
First up is Louis Pasteur. He's the one who actually gave us the name. The word virus literally translates to venom or poisonous fluid. He knew there was some unseen pathogen causing rabies, but he just couldn't see it with the microscopes of his time.
Then we jump to 1892. D.J. Ivanowski.
Ivanowski was intensely studying tobacco mosaic disease, which was destroying crops. He took the infectious sap from sick, modeled tobacco plants, and passed it through a specially designed bacteria-proof porcelain filter. A filter so incredibly fine that it supposedly catches all known bacteria.
And what happened?
The clear fluid that came through the other side of that filter was still highly infectious to healthy plants. This mathematically proved that whatever this new pathogen was, it was significantly smaller than any known bacteria.
Next up, 1898. M.W. Bajerinek.
He essentially replicated Ivanowski's work, but took the thought process a step further. He took that filtered, seemingly sterile fluid and proved that if you rubbed it on a healthy plant, it inevitably got sick. He realized it wasn't merely a static chemical toxin because a toxin would dilute. This thing was actively replicating. So he coined a very famous Latin phrase, contagium vivum fluidum.
Which translates to infectious living fluid. He genuinely believed it was a liquid life form, not a particle.
Exactly. And that belief held until 1935, when W.M. Stanley finally cracked the case. Stanley essentially froze the suspect. He managed to show that viruses could actually be crystallized. And when he analyzed those crystals, he found they consisted almost entirely of proteins. This was the definitive, undeniable proof that viruses were solid, particulate matter, not some magical living liquid.
Okay, let's recap those names. Ivanowski equals the filter experiment. Bajorinic equals the infectious fluid theory. Stanley equals crystallization.
Spot on. Now let's talk about the structure of a virus. Fundamentally, a virus is just a nuclear protein. It consists of two main things: some genetic material and a protective protein coat.
Here's a massive question. Can a virus contain both DNA and RNA at the same time?
Never. Absolutely never. This is a hard, unbreakable biological rule. The genetic material is either RNA or it is DNA. No known virus contains both simultaneously.
And what are the general rules of thumb regarding which type of genetic material they usually carry?
In general, and there are always weird exceptions in biology, but in general, plant viruses almost always contain single-stranded RNA. Animal viruses can be much more varied, containing either single or double-stranded RNA or double-stranded DNA. And bacteriophages, which are the specialized viruses that specifically hunt and infect bacteria, they usually contain double-stranded DNA.
Let's talk about that protective outer protein coat. It's called the capsid, correct?
Yes, the capsid. And it is entirely constructed of smaller, repeating subunits called capsomeres. These capsomeres neatly arrange themselves into either long helical shapes or complex polyhedral geometric forms. Their entire job is to armor and protect the fragile nucleic acid inside.
And the diseases they cause, we all know them.
Unfortunately, yes. Mumps, smallpox, herpes, influenza, the AIDS virus, HIV, all viral. In the agricultural world, plant symptoms are very visual. You see mosaic formation on the leaves, leaf rolling and curling, severe yellowing of the veins, and general stunted dwarfing of the plant's growth.
Next on the unclassified list, we have viroids. These are somehow even smaller and simpler than viruses.
Discovered by a scientist named T.O. Diener in the year 1971. Please mentally highlight that specific year, 1971. It comes up a lot.
He found a new infectious agent that was causing a devastating agricultural disease called potato spindle tuber disease. But this pathogen was structurally different from a virus.
Very different. It was literally just free-floating RNA.
Yeah.
It completely lacked the protective protein coat, the capsid, that all viruses possess. It was just naked infectious RNA, hence the term viroid. And importantly, that RNA was found to be of an unusually low molecular weight.
Okay, naked RNA. Then we get to something that is honestly even weirder and much more terrifying. Prions.
Prions are genuinely the stuff of biological nightmares. They are the agents responsible for transmitting several incredibly fatal, untreatable, infectious neurological diseases. But the terrifying part, they don't have any DNA, they don't have any RNA, they carry zero genetic material.
So how can they possibly be infectious? What actually are they?
They're just proteins, but specifically, they're abnormally folded proteins. Their three-dimensional shape is wrong.
How does a misshapen protein infect an entire brain?
It acts like a microscopic zombie outbreak, or a domino effect. If a pathogenic prion somehow enters your brain tissue, it physically bumps into normal, healthy proteins. And simply through that physical contact, it forces the healthy protein to forcibly misfold into the diseased shape too. It spreads entirely by contact, slowly turning the brain tissue into a spongy, degraded mess.
And what are the major diseases caused by these?
In cattle, it causes bovine spongiform encephalopathy, BSE, which the media famously calls mad cow disease. And the devastating human variant of this is called Creutzfeldt-Jakob disease, or CJD. The prions are approximately the same physical size as viruses, but they act completely differently.
It's truly scary stuff because it defies the normal rules of infection. Okay, finally, let's end this on a much happier, cooperative note. Lichens.
Lichens are a beautiful, classic example of biological symbiosis. It is a highly integrated, mutually useful physical association between algae and fungi.
You absolutely need to know the specific technical terms for the two partners in this relationship.
Yes. The algal component of the lichen is known as the phycobiont. Since it is algae, it is obviously autotrophic. The fungal component is called the mycobiont, and it is heterotrophic.
How does the division of labor work between them? Who is doing what?
It's a perfect evolutionary trait. The algae sits near the surface and prepares food for both of them through photosynthesis. In return, the tough fungi builds the physical structure, providing shelter, and its hyphae actively absorb essential mineral nutrients and water from the underlying rock or soil. They live so intimately intertwined that if you saw a lichen on a tree, you would completely assume it was just one single solid organism.
And they have a very important ecological role regarding human activity, right?
They are incredibly sensitive pollution indicators. They absolutely will not grow in areas with poor air quality, specifically areas with high sulfur dioxide pollution.
So as a general rule, if you see lush lichens growing all over the tree trunks in your neighborhood, you can breathe easy. The air quality is genuinely clean.
Exactly. They are nature's little air quality monitors.
Wow. We have really covered a massive amount of ground today. We started in the absolute chaos of the forest, worked our way through the strict logic of Whittaker's five kingdoms. We looked at the oxygen-proof bunker of the cyanobacterial heterocyst, and we explored the microscopic, indestructible glass houses of the marine diatoms.
It is undeniably a very dense, heavy chapter. But I promise, if you break it down systematically, kingdom by kingdom, and you really focus on understanding the underlying criteria for why things are classified the way they are, instead of just staring at flashcards, it really does start to make logical sense.
Definitely. Just remember to watch out for the examiner's favorite traps. Mycoplasma is a bacteria, but it has no cell wall. Euglenoids are photosynthetic, but they have a flexible pellicle instead of a wall. Deuteromyces are the imperfect fungi with no known sexual phase. And whatever you do, please do not mix up Plasmodium, the slimy protist mass, with Plasmodium, the deadly malaria parasite.
And of course, keep your historical scientists straight. Match the name to the specific breakthrough. Whittaker and the Five Kingdoms, Ivanovsky and the filter, Bajorinic and the living fluid, Stanley and the crystals, Diener and the naked viroid in 1971.
Classification implies order. It is human nature to want to put every piece of nature into a neat, perfectly labeled box. But when we look closely at things like the euglena, shifting between plant-like and animal-like behavior, or we look at viruses hovering right on the jagged edge between being just complex chemistry and being alive, they clearly show us that nature fiercely resists being put into boxes. As the saying goes, the map is not the territory. The biological world is always messier and more wonderful than our attempts to label it.
That is a very poetic truth. But I will add, for the sake of the NEET exam next month, the map is the territory. So you still need to learn the map.
Very well said. Practicality first. So, take this structural logic we've discussed today, go back and carefully review all the specific diagrams in your NCERT textbook, and go absolutely ace that section of the exam. Thanks so much for joining us on this deep dive.
Best of luck with your studies, everyone.
We will see you next time. Keep exploring.